Hey everybody, welcome to week 6. We are going to cover just one PowerPoint lecture today on mycobacteria. So let's just get started. Sharing my screen here. All right. So the objectives are there for you to read through, but overall, again, I'll try to make sure I point out the essential pieces and characteristics of mycobacteria. So in general, mycobacteria are a non-motile rod-shaped bacteria. You'll notice that there's no gram-stain reaction here. I didn't really tell you it was gram-positive or gram-neg, and that's because mycobacteria are they're apart from the other bacteria in that they don't gram-stain properly. And this is all due to that structure of their cell wall. Remember when you first learned gram-staining, you remembered or you learned that the gram-stain was targeting the cell wall. So if your cell wall changes, you can't gram-stain very well. So with the case of mycobacteria, they contain mycolic acids instead of the other normal acetylmuramic acid. It actually is called glycoleomuramic acid, but essentially we just call it mycolic acid, mycolic, kind of get the gist there. It has a very high lipid content, and it just makes it a lot difficult to stain with the gram stain, so it just doesn't take that gram stain up. Instead, we are going to use acid-fast stain for mycobacteria. And so that, again, was also learned back in Micro 1 when we learned gram stain. We learned a little bit about that acid-fast staining process, and we need to remember that for this chapter and for future tests now as well. So acid-fast bacteria are called acid-fast because they resist decolorization by acid alcohol. All right, overall, mycobacteria tend to be very slow grower. It takes anywhere 2 to 60 days for them to grow an auger. There is a group that are going to be what we call our rapid growers, but majority of them are slow growing. There are 2 major groups here. They separate them according to those that do cause tuberculosis, so what we can kind of call our tuberculosis complex. And there are 3 different bacteria that are known to cause tuberculosis. You have, of course, Mycobacteria tuberculosis. And then you also have Mycobacteria bovis and Mycobacteria africanum. The other group is the rest of the mycobacteria, which they just call the non-tuberculosis mycobacteria. So within the Mycobacteria tuberculosis complex, again, there's the 3 listed that cause tuberculosis. The most common cause would be by Mycobacterium tuberculosis. In the good old days, if you will, tuberculosis was always called consumption. So if you ever watch a movie in which the movie takes place many years ago and they're coughing up blood or they're talking about consumption, they're talking about tuberculosis typically. So again, these 3 are all going to be slow growers, and they are also all non-pigmented colonies. And we will get into the different categories that mycobacterium are categorized in. Pigmentation does come into play there, so just keep in mind these 3 are all non-pigmented. All right, a little bit on Mycobacterium bovis. That was one of the 3, of course, that causes tuberculosis. Bovis for cows, it can come sometimes after ingesting milk from infected cows, especially if that milk is unpasteurized. So there is a strain of Mycobacterium bovis called the Bacillus calmette-guerin. I'm not sure how to say it properly, but BCG. And this has been kind of used to immunize susceptible people against tuberculosis. All right, so the whole disease with tuberculosis, it does start with lung. It mimics a lot of different lung infections like pneumonia or a fungal lung infection. Along with that goes night sweats, fatigue, weight loss, and then again, of course, the productive cough with sputum. That's the part that really mimics the other lung infections as well. Now, it can infect other organs besides just the lungs. It will spread. It can go GI tract, lymph nodes, CNS, bone, and joints. So it's a very severe disease. It's not just a little bit of coughing. I mean, it's definitely spreading throughout your body, disseminating, if you will. So people who are immunosuppressed, such as HIV, are very susceptible to getting it, and when they get it, they have a rough time fighting it off. It's very hard on them. So we do have what we call the tuberculin skin test, or PPD, or you can call it the MANTO. God, we love to triple name everything in this life. So the MANTO test is something that every healthcare worker gets done on a yearly basis. It kind of screens you to see if you've been exposed or have tuberculosis, if you will. So again, it's good for just 1 year. all students that go to practicum, well, where I am, and I think it's the same up there, I can't remember, but they have to do 2 of these now. So this is the one where it's done on the forearm, and they basically inject a little bit of the tuberculosis in you. So it's just an extract of it, cutaneously injected into your skin, and then you go back to the nurse about 2 to 3 days later, and they look for a reaction. If you do have a reaction, it tells us that there has been some sort of exposure to tuberculosis, and then they'll do a chest x-ray. If you are negative, then you're negative. And now they used to just require 1 of these. Now they want 2 of these done. So then they'll tell you, come back in a week or 2 and do the whole thing again. So that's kind of what the MANTO test is. As a healthcare worker or future healthcare worker, you'll definitely be doing this on a yearly basis. Your hospital or clinic will give this to you and do that for you. So this just kind of walks you through that whole process of it. All right, as it says here though, by the way, it is not 100% sensitive or specific. That's why they always go to the chest x-ray to confirm, to see if you actually have an infection of tuberculosis. And some people just always react to the MANTO test. They just always have a positive. And if they know that, then they always get a chest x-ray done. They don't even bother with the MANTO on them. They just go to the chest x-ray, because why give them a MANTO if they always have a positive reaction to it? All right, so the other mycobacteria, the non-tuberculosis group, includes all others, and they are present throughout the environment, can colonize us as healthy people, typically not transmitted as much person to person. Tuberculosis, of course, is highly transmittable. They typically, when they're in the hospital, they have a whole special ventilation room that they go into when they have tuberculosis. It's usually the air goes up through a HEPA filter, so that way when you open the door, it doesn't go out in the hallway. But other mycobacteria infections don't need that type of isolation. Now, they are classified into 4 groups, 1 through 4, and they're going to be based on how fast they grow and, again, that colony pigmentation. So groups 1 through 3 are all known slow growers, and then group 4 was the ones that can be considered more rapid growers. So there are your 4 groups. Definitely need to know these 4 groups and why they're grouped this way. So group 1 is called photochromogens. Again, slow growers, and they do develop pigment following exposure to light. So the photo means light, right? So they will develop a pigment after being exposed to light. Group 2 is scotochromogens. They will develop pigment whether it's in the dark or in the light. So either way, they are going to develop a pigment. And again, slow growers. And group 3 is non-photochromogens, basically non-pigmented, again, slow growers. And then group 4 is just those ones that can grow within 7 days. So let's go through some example bacteria that are found within each group. You do need to know the example bacteria in each group and a little bit on them. So within the photochromogens, these are the ones, again, that do develop pigment following the exposure of light. You have Mycobacterium candensii, which comes from tap water, causes a pulmonary disease, and then Mycobacterium marinum. That one's nice because the name tells you where it's coming from. Marinum for marina, like water, salt water. So this is a cutaneous disease. So both photochromogen examples here coming from a water source. Scotochromogens, again, are the ones that can develop pigment in light or dark. You have Mycobacteria salgi. These are hard names. They come from water and soil, pulmonary disease. Strophallaceum is raw milk, water. You see water is a good one here. Pulmonary disease as well as that one, so skin infections. And then Mycobacterium interjectum causes a pulmonary, again, lymphadenitis. Non-photochromogens, you have a really big group here that definitely is really important to remember, and that is the Mycobacterium avium complex. So down at the bottom tells you the avium complex is made up of the Mycobacterium avium, intracellular, and paratuberculosis, lepromyrium, and something they just call wood pigeon bacillus. You don't have to worry about that. The ones that we're really concerned with out of this entire complex is the avium and intracellular. So these are highly infectious for, again, immunosuppressed people like AIDS patients. So they cause havoc on these AIDS patients. They will result in a fully disseminated disease. They might start as a pulmonary infection and then work their way into other organs coming from water and soil. So definitely make note of Mycobacterium avium. Those are very clinically important ones. Mycobacterium ulcerans, water, cutaneous, Zanapi, water, pulmonary, and Melimelonce is chronic pulmonary. And then your rapid growers, you have abscessus, could result in a disseminated disease, pulmonary, that's a little bit more serious there, that one. Fortuitum, post-op infections, and then Killian, cutaneous infections. So I know this seems like a lot, and you're like, oh my gosh, how do you keep all these straight? Unfortunately, all microbiology is like memorization. I don't keep these straight when I haven't looked at them in a while, but the more you look at it, like, you know, we've drilled down Staph and Strep, you know, the more you talk about them, the more you look at them, and the more you work with them, the more they'll dig into your brain, but it is memorization. There's not a lot of easy ways. Think of mnemonics, whatever you can do to help you remember. You'll notice that a lot of it does go with pulmonary or cutaneous, so if you can just try to group together that way. But you do have to remember which group they all sit in overall. So flashcards are a great way. Just try to dig into the material. If nothing else, take away the ones that are the big bad boys like the avium. At least take that away and remember that. So, all right. Let's see. Oh, another one that we definitely need to note that this is significant is Mycobacterium leprae. And just like the name spells, leprae meaning causing leprosy. There is another name for leprosy called Hansen's disease. So if you ever hear mention of Hansen's disease, this is just leprosy. So leprosy is a very much a chronic disease that involves skin, mucous membranes, and nerve tissues. Typically, we're not going to grow this. You're going to look for PCR. So polymerase chain reaction, looking for DNA, that kind of stuff to help identify. Now that I mentioned that, I should say now all of these have been mostly slow growers. I know there is that rapid growers, but the rest have been slow growers. There is, with all the molecular technology that is coming out, and I think I mentioned this back in Micro 1 when we had that 1 week on molecular briefly, there is an instrumentation, and I can't remember, there's probably a couple, but there's one for sure that I know of that I can't remember the name of, but we mentioned it back in Micro 1, that now does PCR molecular, you know, again, using DNA, all that stuff to help diagnose these mycobacteria, especially tuberculosis, because it's such a slow grower. And that would just take, it just takes so long to figure out if that's what the patient truly has. I mean, they have an idea through chest x-ray and symptoms and things like that, but because we want a definitive diagnosis, that molecular machine can pump out a tuberculosis result in 2 hours. I mean, that's crazy. I mean, it used to take days and days and days and days and days just to get the darn thing to grow. Now we can get a result in 2 hours. So molecular is definitely the way to go, especially with mycobacteria because they are so hard to grow and take forever and that kind of thing. All right, specimen collection. Definitely need a biosafety level 2 for most of them. If you think you've got tuberculosis, you better be working with a level 3. So remember, level 2 is the majority of hospitals have a level 2 hood. Those that know that they work with more at-risk, you know, organisms like tuberculosis or brucella or something like that need a level 3, and that's where it's like all enclosed and you put your hands in through like the glove things. Ideally, if we think we have tuberculosis, of course, that is a lung infection first and foremost. So let's get a pulmonary specimen. You can get any of those below listed there. Same with the other mycobacteria. Most of them do cause somewhat of a chronic pulmonary disease. You can grab that. If it's a cutaneous disease, of course, you're going to grab a swab from the lesion or maybe some fluid or tissue biopsy, whatever it might be. There is a way to do fecal samples. This is especially if we think we have a Mycobacterium avium happening with AIDS patients. They can acid fast stain a stool sample looking for that avium complex. And then, again, wound, skin lesion, aspirate. Whatever is being infected is what you are going to collect. So let's talk a little bit more on the acid fast stains. Let's revisit that information. Again, this is for bacteria that have cell walls containing mycolic acids, like our mycobacteria. Ziehl-Neelsen and Killian are the 2 main methods of acid fast, but there is a fluorochrome stain called auramine rhodamine that is also used to help detect mycobacteria. So on that, oh, I thought I had another, oh, I don't have another slide. I thought I had another one. Just make sure that you go back and remind yourself on the Ziehl-Neelsen and Killian. We detailed this out back in Micro 1, I can't remember what week. I want to say it's like week 2, maybe? Week 2, week 1. I feel like it's week 2, but I could be wrong. But there was a whole PowerPoint in which we had it. It's, of course, in your book. But, again, the main difference between Ziehl-Neelsen and Kenyon is Ziehl-Neelsen is called the hot method. It uses heat, whereas Kenyon is called the cold method. It doesn't use that heat. It instead had increased amounts of phenol in it. Remember, it's 3 reagents. Primary is carbofuchsin. Then you have your acid decolorizer, and then your third counterstain is methylene blue. Those that are considered acid-fast positive, like mycobacteria, will keep that carbofuchsin purpley, pink, red, whatever you want to call it, color. Those that are acid-fast negative will keep the methylene blue color. And again, the fluorochrome dye was ormine rhodamine. That should have also been mentioned around the same PowerPoint. And special augers, if we do think we're dealing with mycobacteria, we can use a Middlebrook or a Lowenstein Jennings to help grow those out. So just link these with the growth of mycobacteria. All right, that's it. Just know those characteristics, know the groups, why they're separate, what it means each group name, and then try your best to fit bacteria into the groups as we went through it. So have a fabulous week. Again, if you need anything, please reach out. Thank you.